So this is the second video on key area 1.6. In the first video we had a look at gene mutations. Sorry, there's a bell ringing in the background there. And in this one we're going to have a look at chromosome mutations and at this thing here called polyploidy. First of all, looking at chromosome mutations, well, what do we mean by that? Well, we're talking about a change in the number or the sequence of the genes on the chromosome. Now that's important to remember. So we're talking about genes either moving or being added or being deleted. When it was gene mutations, we were looking at bases being added or deleted. So there are 4 different types of chromosome mutations, and they are deletion, inversion, translocation and duplication. Now the only one that's got the same name as your genetic mutations is deletion. The other ones have got these different names, and I've put a wee short mnemonic here, DICT, which stands for deletion, inversion, chromosome, translation and duplication. If you can think of some other way to help you remember that, then do it. Or sometimes what I do to help me with remember things is I make up some sort of wee sentence for the name that is here. So dogs in China try, I don't know, dynamite or something like that to help me to remember. So if you can think up some sentence that helps you to remember the types of chromosome mutations, then you can do that. So let's look at the first one, which is called deletion. Now deletion mutations have a drastic effect on an organism because you're basically losing genes. And here's an example here where this part here of the chromosome, a number of genes have been lost. An example of a deletion mutation is one that is where part of chromosome 5 is missing, and that leads to a syndrome called the Cry-du-Chat syndrome, and that is, or Cry-du-Chat, my French isn't that great, and Cry-du-Chat is meaning the cry of a cat because it's a child that has got this condition cries as if it's like a cat, and you can always look that up on the internet and you can hear the cry. Next type of chromosome mutation is this one here called inversion. Now what that means is that where you've got a normal sequence of genes, so here if we say this is the normal sequence of genes here, what happens is that sequence gets inverted, it gets turned around. So this gene ends up down here and this gene ends up here. And what happens with that is that you end up with what are known as non-viable gametes. Now gametes, if you remember, are the sex cells, and gametes are formed from a type of cell called a gamete mother cell, which has 2 sets of chromosomes, and what happens is the gametes are formed by the matching pairs of the chromosomes lining up, and then there's a separation process that takes place. It's a process called meiosis. You don't need to know that at higher biology, but what you do need to know is that the matching chromosomes, or what happens in inversion is that those matching chromosomes have trouble actually matching up because the genes have been inverted. A non-viable gamete means gametes that aren't actually going to go on and produce fertile offspring. They can't be fertilized by sperm. So here, translocation. Now translocation involves the transfer of genes from one chromosome to another, and that also results in the problems with the pairing of chromosomes during gamete formation, and also leads to non-viable gametes, so gametes that can't be fertilized. And here's some examples of translocation. This type here is called non-reciprocal translocation. And what happens here is that you'll see here's a chromosome here, chromosome 4, and here's chromosome 20. And what happens is that a piece of chromosome 4, this piece here, ends up in chromosome 20. So you end up with a situation like this, where there's an extra number of genes here in the chromosome 20, and there are some genes that have been lost here from chromosome 4. So that's non-reciprocal translocation. Now and it's called reciprocal translocation. So all that means is, well, say in this situation here, the chromosome 4 here, part of it ends up at the end of chromosome 20. But what happens is that a piece of the chromosome 20 ends up on the end of chromosome 4. So they're kind of, I suppose you could think of it as swapping some genes between each other. And that's why it's called reciprocal. Lastly, looking at duplication, well, this results in a set of genes being repeated, and some duplications can be detrimental, and an example of that is oncogenes. Duplication of oncogenes is a common cause of cancer. So here you'll see, here's a section here, and what has happened is that section has been duplicated on the chromosome. Now, I've said that some duplications can be detrimental. Some duplications can be a good thing. And it's important when we think of this in terms of evolution, and we think of this in terms of natural selective pressures. So when a gene is duplicated and you get a second copy of that gene, you've already got the first copy of the gene, and it will be subject to selective pressures. So say that gene is something that is good for the animal in a certain environment, and then the environment changes and it no longer becomes a beneficial gene, then there's selective pressure on that gene. But the thing is, if you've duplicated that gene, so you've got the two genes, here's one here, here's the second one, the duplicated one. This gene here is the one that will be subject to selective pressures. So say it's a gene that gives you some sort of ability to survive in, say, very, very warm climates. Well, this, when it starts to become colder, then this gene is no longer advantageous, and it could be that the organism will then die. So, but the thing is that this second gene that's here, it isn't affected by any selective pressures. So it can go on mutating, it can mutate to produce new DNA sequences that are in there. So say, imagine this has been deleting away there, and it actually, and the example that I've given here is a fish that's got an extra gene, which produces a glycoprotein that acts as an antifreeze. Now, obviously, in hot weather, that's not of any importance, but if it starts to get colder, then this antifreeze that's being produced is a beneficial thing. So that's why I said before that some duplications are a disadvantage to an organism. They can be cancer producing, but other duplications can be good, and that gene can go on and mutate free from selective pressures, and it can confer some sort of advantage which will possibly increase the chance of the organism's chances of survival. So that's chromosome mutations, and you have to know all those mutations and be able to tell the differences between them. We're going to go on and have a look at this special type of duplication of chromosomes, and it's called polyploidy. And what happens with that is it results in an extra set of chromosomes, and it's the result of an error during gamete formation. Usually when gametes are formed, you've got, say, I'll put in here, matching sets of chromosomes, and usually what would happen is when the gamete is formed, you would end up with gametes with half the number of chromosomes. So what happens is just one of these chromosomes ends up in the gamete. But if something goes wrong in that process when division takes place, you could maybe end up with an organism or a gamete which has got, say, these two pairs end up in the gamete and just one of these chromosomes end up in the gamete, or there's all sorts of other arrangements. So you'll see that In this case here, there's 3 chromosomes present instead of the 2, because in this original one we had 4 chromosomes. The gametes would have the number 2 chromosomes, but this one here, something has gone wrong during the gamete formation, and you end up with 3 chromosomes. And when that goes on and is, say, fertilised, it could be fertilised by a normal sperm gamete, which has just got the 2, but you end up with an individual, when fertilisation takes place, with 5 chromosomes at the end of this process. Whereas, if fertilisation of this gamete was occurring, this one here, you would end up with an individual that had 4 chromosomes. Now, polyploidy is very common in plants. I'm saying roughly 50% of plant species are polyploid. Polyploid plants are normally larger and they have an increased seed and fruit size, and that obviously economically. So here, for example, here there's some polyploid strawberries that are here, and you can see that they're much larger than, say, a non-polyploid strawberry. And farmers use that to their advantage, breeding plants that have got polyploidy, because they'll get larger fruits. If you've ever seen a strawberry out in the wild, they're actually really quite small and not particularly tasty. So polyploidy is something that's used, or something that happens just naturally, but it can be used in a breeding process to produce fruits that have got higher yields. Polyploidy plants with an uneven number of set of chromosomes are sterile. So if you've got ones that have got 5 chromosomes there, or 9 chromosomes, it means that it's sterile. And an example of that would be bananas, which is you produce a seedless fruit. Now here's an example of what I was talking about before in the previous slide. This is a formation of a triploid zygote, where there are 3 copies that of the chromosomes that are extra copies that are produced. And you'll see here, over here, this is normal gamete formation. So here we've got what is known as a gamete mother cell before the gametes are formed. It's got 1, 2, 3, 4, 5, 6 chromosomes in total. And when the gametes are formed, each of the pairs end up in a separate gamete, and you actually end up with 4 cells. There's a reason for that, but we're not going to go through the meiosis process. So you end up with gametes. Here's one here, a gamete which has got half the number of chromosomes, 3 chromosomes. Now normally that would fuse with if it fused with a normal, another normal gamete, you would end up with an individual that has got 6 chromosomes. But in the example over here, an error occurs during gamete formation, and the chromosomes here, they don't separate. And so what happens is this abnormal gamete is produced, which has got still 6 chromosomes in it. So therefore, when that gamete fuses with this other gamete, you end up in total with 9 chromosomes, because you've added an extra 3 chromosomes to the cell. Now, it's pretty rare for polyploidy to occur in animals, but it is thought that polyploidy contributed to the evolution of vertebrate animals. That there's some contribution in there, but mammals which have polyploidy usually fail to survive. Now, there are exceptions to that, and an example here is this rat here, which is found in Argentina, and it has a double set of chromosomes, except for in the gametes. The chromosomes there aren't double. And that is a summary of chromosome mutation and polyploidy. In addition, this is the end of Key Area 1.6, so you should make sure that you've read through your textbook as well, add some added bits of information, and also make sure that you've completed up to topic 6 on your Scholar. I'll also have some videos are posted on Key Area 1.2 that you can watch too. Sorry, I meant to say Key Area 1.6.